So today's topic is going to be on lipids. So this is more of a review for you guys, and then I'm going to a bit more detail than it is. So we're going to go into lipids. So just a reminder, lipids are basically a nonpolar hydrocarbon, right? They don't really, in other words, they don't really mix well with water. They're nonpolar, right? So it's just a bunch of carbon chain with hydrogen. And there's 4 types of lipids I want you to recognize. One of them is going to be triglyceride, phospholipids, steroids, and then wax. And I also want you guys to understand how later on, starting tomorrow, I want you guys to understand how triglycerides are broken down as well as synthesized. Same thing for wax. I want you guys to understand how wax are synthesized as well. Okay? So let's go over those together. First thing is first, I go into triglyceride. Again, today is just going to be introduction. I'm just introducing you again to the lipid topic. So most of this will be a review for most of you guys. All right, so triglyceride. Remember, this is triglyceride example here. And it's made up of a glycerol head. And you guys remember glycerol in last exam? Glycerol enters the the glycolysis cycle to become glyceraldehyde 3-phosphate, right? You remember that? So that's what glycerol and the purpose of glycerol is. It's just a 3-carbon chain. 1, 2, 3. So that's glycerol, and plus 3 fatty acid tail. All right? And we're going to talk more about this fatty acid tail specifically tomorrow and what purpose it could do and what significant power these fatty acid tail has. So triglyceride is another form of energy source, and it's mainly stored in your adipose tissue. If you eat a lot and your body have no idea where to store the excess fat, they're going to store in your adipose tissue, right? Your gut. That's basically what it is. But the thing about triglyceride is that it gives a lot of energy. Once you break it down, it's going to give a lot of energy, a lot more than 1 glucose, which is insane. So starting tomorrow, you're going to recognize and realize how much energy, just cutting this little small piece out, how much energy that could give you in comparison to glucose. It's a lot. It's insane. All right. So triglyceride, and this is another thing I want you guys to recognize as well, is this 2 terms, saturated fat versus unsaturated fat. So let's go with this one first. In regards to saturated fat, you can think of this way. When your hydrocarbon tail, your fatty acid tail, is saturated, the term saturated means it's saturated with hydrogen. So in other words, it's just filled with a bunch of hydrogens. Like all the carbon is hydrogen everywhere. Okay? So that's what it means by saturated. I mean, you're saturated with a bunch of hydrogen, meaning there's not enough space to make a double bond. So it's basically all single bond if it's saturated. Okay? And typically, we could find saturated fat more in animal cells, like ourselves. Animals typically produce more saturated fat. And the huge advantage of saturated fat, although we claim it to be unhealthier, because plant oil is more unsaturated, but animal tend to be saturated, like butter, right? Butter is saturated. In room temperature, it's solid, right? But saturated fat do have a huge advantage. That huge advantage is that it has more energy, and you will learn that tomorrow. Why saturated fat gives off a lot more energy than unsaturated fat. Ironically, it's unhealthier for some reason, but the unhealthiness actually comes from it being tightly packed. You could think saturated fat, when a cell is tightly packed together, it creates a more stronger barrier, preventing water from passing through. So in other words, it's disabling the cell to breathe easily. And saturated fat, just like cholesterol, it could eventually lead to something called heart attack, right? Because it's become very sticky, and and eventually it causes. I'm not sure. I talk about cholesterol, how it keeps it's your blood vessel, for example. Let me draw it out really quickly, just a reminder. Imagine this is your blood vessel, and with cholesterol as well as saturated fat, because it's just very sticky, it's going to eventually build up and become very sticky on the membrane. And if it becomes sticky like this, imagine I'm just drawing unsaturated fat, saturated fat and cholesterol. it creates a high pressure in one side. And it creates this kind of high pressure. First, it leads you to high blood pressure, and that will potentially lead to a burst. In other words, a stroke or a heart attack, right? So that's a huge disadvantage of why it's unhealthy to eat a lot of saturated fat like butter, right? But it does give you a lot of energy though. But again, it prevents you from breathing easily and so forth. So again, that's saturated fat. So again, just a summary, if you're wondering what saturated fat is, there's no double bond, there's no kink, it just goes straight down, and it's saturated with all hydrogen. All right. The next one here will be unsaturated fat. Unsaturated fat is typically found in plants, right? And usually it is liquid in room temperature. So like vegetable oil, olive oil, right? Those are basically all unsaturated fat. Now these guys, they have more ability for locomotions. So they're less packed, allowing for more motion, which is why you see them as liquidy form in room temperature, because they're able to be fluidity, right? Which is pretty good. Having the ability to have easy movements and all that, it makes it easier for molecules to transfer back and forth between cells. So it's able to breathe easier, able to transport molecule easier, which is always good. And keep in mind that unsaturated fat are always in their cis format, not trans, okay? So cis, what does it mean by cis? Cis means same side. So, and remember, unsaturated fat has a double bond. So how do you determine whether it's unsaturated versus saturated? Unsaturated fat, so long there's a kink, like this, those are unsaturated, which gives more space for movement. And that kink is created by a double bond. You see this double bond right here? These are what makes them unsaturated. But there's a difference here. Unsaturated fat is always cis, same side. So when you're creating it, it looks like this. See? One is going downward and going downward, the same side. Trans is opposite. So one is going down and the other one's going up. Please try your best to recognize the difference between trans fat versus cis fat or unsaturated fat. Okay? So make sure you guys do your best to recognize that. So this here is going to be unsaturated fat, cis, okay? All right. Now, I did briefly mention trans fat. What exactly are trans fat? Well, trans fat are basically unsaturated fat, but they hydrolyzed it. They hydrogenate it so that it becomes trans. Supposedly that there's a reason why. They usually find this in like factory chips for preservative, and our body are not able to break down trans fat easily. We have a very difficult time breaking down trans fat. So every time you're eating trans fat food, those are literally stored in your body for much longer period of time, and they cannot break it down. Even if your cell's trying to break it down because you're starving, they have a very difficulty breaking it down, and so you actually become more tired. So trans fat is not really healthy because your body is not adapted to break down a trans hydrogen bond. They're able to break down cis double bond, but not trans double bond. Okay, so those are three differences between these guys. Okay, any questions so far? All right. So just a reminder, where exactly this triglyceride goes to, like especially glycerol. Just a reminder. This glycerol is 3 carbon, 1, 2, 3, and it enters glyceraldehyde 3-phosphate. You guys remember that? And here's the full mechanism that I want you guys to recognize here. So glycerol, it will become hydroxylase, just added a bunch of OH to them. So 1, 2, 3. So 1 OH is removed. So glycerol contains 3 OH group, hydroxyl group. There's carbon, hydroxyl, so the carbon and then OH, carbon, OH, carbon, OH. Make sense? So all these 3 carbon contain an OH. Once it undergo glycerol kinase with the help of ATP, well, this carbon, the OH here, disappears because it binds to ATP instead, one of the phosphate groups. So therefore, now it becomes glycerol 3-phosphate. This guy, the third carbon here has a phosphate head, it does not have OH, while the other two still have OH. Right? So in other words, it loses an OH and becomes this. Okay? And afterwards, it undergo another enzyme called glycerol 3-phosphate dehydrogenase, which utilizes NAD to become NADH, which is very important for the ETC cycle. But once it undergoes this, the middle carbon loses its OH. Okay? So now we only have one OH left. And at the end, undergoing some unknown mechanism, it becomes glycerolaldehyde 3-phosphate, which now they don't have any more OH. Okay? So basically every single step is just removing an OH from those carbon. Okay? If you're wondering what it should look like, let me just draw it out for you guys just in case. When I talk about OH, this is what I meant. 1 second, screenshot. OH. This is actually what glycerol is. OH. That makes sense for you guys? It just have a bunch of OH. Like this. So carbon attached OH, and every single step is just removing that OH. Make sense? So that's basically what's going on in these 3 steps. So step 1, glycerol kinase, remove one of the OH. Okay? The next step, glycerol 3-phosphate dehydrogenase, remove another OH. Right? And then the last step, to become glycerol 3-phosphate, it removes the last OH. And done. That make sense? So that's how it becomes glycerolaldehyde 3-phosphate. It's just removing those OH group. Okay? Yes?
So what about lactic acidosis?
What's that?
What about lactic acidosis?
Oh, let me go over that. This excellent question. All right. So, okay. So this is going to be 1 or 2 questions on the exam or a quiz. It's going to go on over the drug called metformin. Metformin. You guys heard that drug before? It's a diabetic drug for type 2 diabetes. It's very commonly used, right? This particular drug here plays a big role in inhibiting the ability for glycerol to enter the glycolysis pathway. It specifically inhibits these 2 enzymes, glycerol kinase and glycerol 3-phosphate dehydrogenase. And when this occurs, it prevents the ability for you to make more glucose, right? But at the same time, it also prevents for you to go through another cycle. So it prevents you from going both pathway, glycolysis and gluconeogenesis. It prevents those 2. And therefore, what's going to happen is, if you take the drug a lot, one of the side effects, there's side effects to the drug, but one of the side effects will lead to something called lactic acidosis. The reason why is because you're preventing it from going, basically you're affecting this whole entire glycolysis pathway. You're now shifting the equilibrium to where there's going to be more lactic acid being created because you're affecting this particular pathway. But the reason why people use this drug is mainly to prevent more glucose from being made, because you want to reduce the glucose level in your blood. So people take this pathway. But when you do that, it increases the equilibrium. It shifts the equilibrium to where you're making more lactic acid. And that's what's going on here. Again, metformin is a drug that affects these 2 particular enzymes. And that's a common diabetic drug. All right. And also keep in mind, this particular pathway only occurs in the liver or kidney, and kidney. Sorry, liver and kidney. So nowhere else. Yes.
What would we call this pathway? Like, how would you refer to it?
Oh, this is glycerol catabolism. You're breaking down glycerol into energy. All right. Cool. And another form of lipids. Oh, that was triglyceride that we went over so far. So any question about triglyceride? Wow, that was a lot for triglyceride. So long you understand, how glycerol is broken down, and you understand about saturated, unsaturated, and trans fat, I think you're good so far. That's what we covered so far. So you guys good? All right. The next one we're going to cover will be wax. I'm going to briefly cover this because we're not going to focus too much, but just keep in mind that every single one of us have wax. I mean, example of wax would be like the cuticle of leaves. That's waxy layer. Like it's waterproof, right? On our skin, we also produce wax ourselves, right? So if you're wondering, do we produce wax? Yes, we do, right? Feathers also has waxy layer. So how exactly are wax produced? Well, later we'll go into more detail, but this particular guy right here, this is a fatty acid from the triglyceride, right? This is triglyceride, one of the fatty acid pieces from the triglyceride. And if this, instead of going to create energy, like you break it down through beta oxidation, which we'll talk about tomorrow, instead of going through there, we use that and undergo something called acyl-CoA reductase. So if we reduce it, we actually convert, instead of turning out to energy through oxidation, if we reduce it instead, we'll turn that into wax. So it could go through either end. You could get oxidized into energy or get reduced into wax, which is a protective layer for our skin. Right? So energy or protection, choose one, right? So if it does go through reductase, then it will become a wax. So it goes through reductase twice. So acetyl-CoA reductase, and then acetyl-CoA reductase again. And then at the end, you have CoA ester, become wax. So in other words, in order to become wax, it utilizes mainly one enzyme, acyl-CoA reductase. And it undergoes that same cycle twice. Okay? All right, the next set of lipids that I'll briefly go over, not too much, it's going to be steroids. Now, steroids, you guys kind of already know it, but the biggest steroids I'll talk about here, besides, sex hormones and the stuff we talk about, like make you stronger, this one, the biggest hormones I'll talk about is going to be cholesterol. That guy is a steroid. And especially that guy is the parent of almost all steroids that you know, almost all hormone. Why? Because cholesterol becomes many other hormones, such as vitamin D, such as estrogen, and then estrogen will become androgen, which is actual testosterone, right? So it's basically a parent structure of many other hormones. And hormones, something very unique about hormones, they have a very unique property where they're able to seep into skin. So in other words, if you were to rub hormones on yourself, you may think it's not going to affect you in any way. However, because of its unique shape and property, it's able to soak into your skin pretty easily and be absorbed and produce something called cell signaling. So you got to be very careful when you're dealing with hormone. It's not through needle shots that you need to stimulate your body. It's just by touching with your bare finger, and that's enough to go through your skin to stimulate some sort of response. So you got to be aware of those kind of steroids, okay? And lastly, but not least, and today's topic, we're mainly going to be focusing on phospholipids. And then tomorrow, we'll mainly focus on triglyceride. So our main topic starting today is going to be all on phospholipids. So what exactly are phospholipids? Well, first things first, what they're composed of, they have a phosphate head, PO4, right? Phosphate head, and two fatty acid tail. That's basically what it is, a phosphate head and two fatty acid tail. And they're linked together, if you were to talk about their linkage, it's through something called phosphodiester bond. Very quickly, let me go back all the way up here. This is what we call an ester bond between glycerol and your fatty acid tail. That's because it's just basically OH that's interacting. So OH group interacting with a carboxylic acid, you call that an ester bond. For if you guys not yet taken OChem, don't worry. Just know that glycerol in order for glycerol to link with a fatty acid tail, they need to create something called an ester bond. Okay. On the other hand, phospholipids, they don't have a hydroxyl. Rather, it has a phosphate. So we call it phosphodiester bond instead. Phospho because of phosphate, and then di because there's two of them, and it's linking with a carboxylic acid from the fatty acid tail. Therefore, we call it ester. So phosphodiester bond. Okay, but for those who never taken OChem, just know that because of the phosphate head, we call it phosphodiester bond. Okay, and phospholipids plays a big role in your cell membrane. It acts like a skin. It's a barrier between the environment versus yourself, right? It's like a waterproof barrier, which is really nice. I mean, the last thing you want is anything to go into your body. That's pretty dangerous. So having the phosphodiester bilayer is very important. And having this kind of bilayer is what we call phospholipid bilayer. It's a bilayer that we see. Okay? And it plays a very big role. So internally, this bilayer is all hydrophobic. Externally, and as well as intracellular, it's hydrophilic. Loves water, hates water. Because we have both barrier, it makes a very strong barrier, right? So, pretty nice. Another thing about this kind of membrane is something very unique I want to mention as well. So whenever you're cold and shivering, and same thing, and you've seen this before, when we talk about the concept freeze and thaw, whenever you freeze something or make something very cold, the membrane becomes almost rigid, like frozen like literally frozen, right? It's become tightly packed and rigid. So when it's very cold. And then when it's very hot, then the gel, this phospholipid bilayer is become more flexible. So this is when it's very cold, they become very rigid, stick together, like compressed together. And when it become very hot, things want to move around more easily. But that's mainly because the bilayer is trying to breathe, right? So it's so hot, you're trying to breathe out air. That's why it's able to fluctuate. And that being said, because of their ability to be fluctuated like that, in reality, I'm not sure you know this, but lipids, every single phospholipid bilayer, they don't stay in one spot. They're able to move around anytime they want. They can move back and forth and so forth. So this same lipid right here, this same phospholipid, it's not just here. It can move around the circle, do a zigzag, anywhere they want. It's fluidity, right? It's able to move like water molecule. But the thing is, they can't really flip to the other side. So this up here, this phospholipid in the extracellular, cannot become intracellular. However, the only way they could become intracellular or extracellular or vice versa is through the use of a protein called flippase. So they require a protein called flippase in order to flip them from one side to the other. So in other words, with the help of flippase, now the phospholipids could literally go anywhere they want. Well, so long within that same cell. All right, that's all the lecture I have. The rest is going to be on the topic on diffusion, osmosis, facility transport, and active transport. Okay. Before I move on there, I'm going to give you guys a 2-minute breather. So go ahead and breathe. So you guys already did this before, but I want you to do it again. So first things first, everyone leave their stuff on the table, and let's do this. Let's have everyone here gather where I am really quick. Hopefully you guys still remember this analogy. Oh no, what you want to continue. When you think is relevant, just clip it off. So let's do this. All right. So I want everyone here to gather where I am, and I want you to cluster to be and make this area a very high concentrate of students. All right. Now I want you guys to go ahead and diffuse back to your seats. Cool. Wow, you guys did it. Diffusion. All right. All right. So that's what you did. You see that? You did something called diffusion, where you guys gathered from a a very high concentration of students, or in this case, of substance, to a very low concentration area, right? So you guys kind of spread out to where everything is now evenly distributed. Very good. And we already mentioned, what affects diffusion, right? So we don't need to go over the detail. So again, temperature, concentration, size, charge, pressure. You can come up with any reasons, and it could speed up diffusion or slow it down, right? And the nice thing about diffusion is that it doesn't require ATP. So it just happens. So you guys decided to just take a seat yourself, right? So I don't need to bribe you at all. All right. The next topic I want to go into now is going to be osmosis. And again, you guys already did this, but I got to have you guys get up again. Keep exercising. Let's go, guys. We need to keep exercising. So if everyone here, go ahead and get up and gather to where I am again. Now this is going to be slightly different from diffusion. So, as a diffusion, you guys assume that you guys are clustered in a high concentration here, right? But, let me take this for example. So diffusion is high concentration of solute, which is you guys, going out to lower area, so evenly distributing around this whole entire environment. Osmosis, on the other hand, is slightly different. Your goal, now let's assume instead of solute, let's assume you guys are solvent. So you guys are water rather than salt. You guys are water at this point. And your goal is to take a seat. And there's only one seat here. Did that make sense? I don't think all you guys could take a seat here, right? There's no way. So there's a very low concentration of solute. But there's a high concentration of solute out there, right? So osmosis is basically the travel of water from a low concentration of seats to a higher concentration of seats. And that's the main difference between osmosis and diffusion, right? So osmosis is you guys as water trying to find your seats. And there's only one seat here. So go ahead and osmosis back to your seats. All right. All right. So before we move on to the next topic, what I want you guys to do now is, can I have you three over here? Go ahead and take a seat at this table, and bring all your stuff. And then you guys have a lot of students. You guys good? I want you guys to go ahead and discuss among your table the differences between diffusion and osmosis, and go ahead and clarify to each other. Yes, sir.
Does osmosis require ATP?
No, none of them require ATP. So go ahead and discuss among you to ask for clarity the differences between osmosis and diffusion.
Does he know why? Yeah, he's okay. He gets them. So let's remember.
All right. Let's do a quick exercise with one another, okay? So, what I want you guys to do now is, I want you guys among your table, or actually everyone here, take out a piece of paper. If you have, or your iPad or wherever, it's fine too. But just draw 3 boxes. 1, 2, 3. And then within those boxes, draw like a circle right here, right? Like this. And then write down these numbers. 5% salt, 1% salt, and so forth. So go ahead and draw 3 boxes. And then within those 3 boxes, 1 circle, all 5% salt. And outside here is 25. So draw exactly what I see here. And then I want you guys to discuss among each other for the next 1 to 2 minutes. Will water, based on what you learn, where it needs more seats, would water move into the cell? The cell is the circle box, or out of the cell into the red box. All right. All right, let's go over this together. So the first question, the first one, will water go into the cell or outside the cell?
Into the cell.
The water will go into the cell, right?
Yes.
There's higher percent of tables or seats here, right? So that's water, you prefer an area that has more seats. That makes sense? What about the middle? Inside or outside the cell? Stays equilibrium, it stays the same. Very good. And the last one?
Outside.
It will leave the cell, right? Because there's more percentage of table out there, or seats out there. So you guys kind of make sense in regards to osmosis? So hopefully it's not too bad. Try your best to understand it, okay? All right, but there has to be a name given to them. And there is. We're going to do this. The one that has low salt and the one that has higher level salt. the one with low salt, we can call that hypotonic. So because the environment is low salt, we'll call that hypotonic environment. Hypo. Hypo, low, right? Po, low, right? So hypo, low, polo. And then the other one inside a cell here is very high salt. And so they're pretty hyper. When you have your high level of sugar, you're hyperactive, right? So in this case, this guy's very hypertonic. If they're equal, we'll call them iso. Both the environment and the cell, they're both isotonic. On the other hand here, the last one, because there's higher level of salt in the environment, we'll call it hypertonic salt. It's hyper, right? Hyperactive, a lot of salt. On the other hand, the cell has very little salt. So because they have very low salt, we call it hypo. All right. So you guys understand the terminology to what's going on there? So terminology to the level of salt. If you guys understand that, we can move on. But before that, any questions so far? You guys solid? So hopefully you guys learned this in the past before. Yes, sir, man.
So when we use those words, we can refer to either the cell or the environment?
Correct. So you can call it hypotonic cells or hypotonic environment. Correct. But you have to refer it to something. You can't just say hypotonic, otherwise I have no idea what you mean. Correct. So the main question here is, what exactly are the consequences of the osmolarity? If you were to put your cells in water or put your cells in salt, what would happen? So what I want to do now is give you guys the next 2 minutes, discuss among each other the consequence, the osmolarity consequence of these three. All right, ice glacier melting into salty water, what happened? Forgetting to add any salt in an IV bag, what will happen? And leaving a potato slice in salty water, what happened then? All right, so any volunteer shout out is fine. So what's the consequence of ice glacier melting into the ocean? What do you guys think? What's the consequence? Decrease the salt concentration in solution. What will happen to organism that's living there? Right. So water will flush into them, so they start poofing up and they may die, right? So that's very dangerous, right? If a bunch of water poofing into your body, your cell will start exploding and you go through hydrolysis, which is scary. That's actually how we lyse our cells, is through hydrolysis, right? It's lysing plus water, which is very scary. That being said, what about forgetting to add any salt in an IV bag, which becomes 0% salt into your body? What will happen to your patient?
They shrivel up, become hypertonic.
What do you think?
Cells in their body will nice.
The cell in the body will also it will bloat up. All the water will rush into the cell. In our body, I'm not sure you guys know this, but in our body, the salt content is about 0.9% salt to 1.2% salt. So we do have some sort of salt level in our body. It is very important that we maintain it around there. If it's higher or lower, it could be very deadly to your body, especially to your kidney. Because your kidney can only filter out so much. And if you were to dilute out the salt from your blood, your body's going to go through a lot of explosive death, right? Because now all your cells will start exploding and dying. And it's very scary, because imagine you're a nurse or you're a doctor and you're working with a nurse that has no idea about the importance of salt. You're going to be liable for that patient that you just killed. Even though it's not you, but you indirectly caused it. That's pretty bad, right? So you got to be very careful. And remember to add some salt in that bag. Okay? And what about leaving potato slices in salty water? What happened to those guys? Oh, that's right. I knew I'd forget something. I was going to do a potato slice example for you guys, where I have one in DI water and the other one in salty water. But if you put in salty water, what happened to potato slices? It would shrivel, right? It become all mushy. It will kind of be, because potato slices, they're pretty stiff. But once you put in salt water, all the water from the potato will flush out and it will shrivel. Therefore, when you pick it up, it's able to bend and flex so easily without breaking. That's what happened in potato slices. That being said, you could do the opposite. I'm not sure you ever. One of the biggest secret ingredients of making crispy French fries is to leave it in DI water, so that it become crisp, right? So if you ever gonna be making your own French fries in the future, leave it in DI water or just regular tap water for like 20 minutes or overnight. It's up to you, and then you can cook it at the end. It becomes very crisp. All right. So those are some applicational consequences of osmolarity. But I'm going to show you one more video of the consequence from osmolarity, okay? And this here, yes, there's going to be 1 or 2 questions on the quiz or exam on it, so make sure you guys pay attention. And there's also application as well.
Go. Your go-to solution for getting quality work done quickly.
There's a time.
All right. So, have you guys ever heard that you should never give baby water or never let them drink water? And there's a reason behind it. And that reason will be osmolarity. All right? So, let's watch this video together.
In every person's life, when drinking water is deadly. Even as much as a few ounces is enough to kill. The average healthy adult is made up of 55 to 60% water. The average baby, on the other hand, is roughly 75% water. And that difference is why babies shouldn't drink water before they're at least 6 months old. Not from the tap, or a well, or a spring. Plain water is just plain off limits. You see, everyone's kidneys have a limit to how much water they can handle. Break that limit, and the excess water will back up into your bloodstream, where it dilutes the salt, or sodium, in your blood. Once your blood dips below 0.4 ounces of sodium per gallon, you're at risk of a condition called hyponatremia. It happens when your cells try to return sodium levels to normal by absorbing the extra water, and swell up like a water balloon in the process, causing complications like confusion, vomiting, and muscle spasms. Hyponatremia is common in marathon runners who drink too much too fast during a race without also providing enough sodium to balance out their blood. And if you keep drinking, that excess water will eventually reach the cells in your brain. By this point, you have a dangerous case of water intoxication, which affects roughly 200,000 Americans each year. It occurs when your brain cells swell, which builds up pressure inside your skull that can lead to seizures, brain damage, and in severe cases, death. But don't worry, dying from water intoxication is extremely hard for a fully grown human. An adult would have to drink 2.5 to 5 gallons every few hours to reach that point. But for newborns, it's a different story. Their kidneys are about half the size of an adult's, so they can't hold much water to begin with. And it takes just a few ounces to cause problems. On top of that, their kidneys aren't developed enough yet to properly filter water, so any water that enters their body ends up in the circulatory system, where it dilutes their blood and increases their water content by 7 to 8%. But it's not just drinking straight H2O that poses a threat. In fact, most cases of water intoxication in infants don't even involve a glass of water. A common mistake is when people dilute baby formula too much by accident, or when parents dunk their infants up and down in a swimming pool, and in the process the infant gulps in too much water. It's important that if your baby is showing signs of water intoxication, that you take them to the hospital immediately, where a doctor will likely provide some form of fluids, like intravenous saline solution, to bring the infant's sodium levels up to normal.
You guys learned something there? All right, so that was diffusion as osmosis. The last 2 topics we'll go into now will be facilitated transport and then active transport. All right, let's go over this. Oh, okay, this activity. Cool. But before I move on to this activity, guys, go ahead and finish up taking your notes. And then once you guys done, let's do this. So what exactly did we just went over? So facilitated transport, what exactly is that? It's basically when there's a protein channel that allows large molecules such as sugar or other molecules like drugs, proteins, enzymes that's important and relevant to enter the body. And that protein is also very, very specific. They don't allow just any protein to enter. They allow a very specific protein to enter. In this case, I only allow students who has shown proof of their ID to enter, right? And only they're able to enter. The rest of you guys are stuck out there, and we just close the door unless you have an ID, right? And that's basically it. So again, the same concept where everyone here is gathered outside, very high concentration, but you eventually fluctuate from high concentration to low concentration, right? But it's very specific manner, okay? So that will be here, facilitated transport. Now, on the other hand, and keep in mind that facilitated transport does not require ATP. On the other hand, active transport does. Active transport does require ATP. And an analogy for this one is for, let's just say, the remaining students out there, and there's already like an equilibrium of students here and inside and outside. So in order for the student outside to enter, they had to give me some money. Or something like that, right? Along that line. They had to spend some energy in order to get inside. And it's basically them moving from a very low concentration of solute to a more higher concentration of solute. So in other words, we're trying to force molecule coming in against equilibrium, against diffusion, right? And there is mechanism that does that. For example, calcium and potassium ion channel flux definitely require active transport, and they play a big role in our muscle and neuronal chemical signaling. So again, here's an example. Sodium and potassium ion channel flux. They're very important for our organ and neuron to function, so that we're able to stimulate our brain and communicate with one another in our brain. And there's also something called calcium ion channel flux. This one here is controls and help with the muscle function, allowing you guys to move using ATP and so forth, right? So active transport plays a big role. It is against the gradients, but they play a big role allowing you to function, because without calcium transport, your body's going to be very stiff and you could barely move it. Like, your body is literally stiff and you could barely move it without calcium. And so I'm not sure you've ever seen like an animal that recently died. You'll know they recently died because they have so much stiffness in their body, you can barely move them. That's because the calcium flux got disrupted. All right. Now, due to that reason, because cells requires calcium and sodium and potassium to be in a very high concentration against the equilibrium, against the concentration gradient, against diffusion. Because of that, we actually could develop a drug to utilize that particular mechanism to kill other bacteria, infection, and other diseases. So one of this drug is a form of ionophore antibiotic. Ionophore antibiotic, what it does is that it looks like this. This is an antibiotic. And it goes into the bacteria, and it causes a pore in the bacteria. When it causes that pore in the bacteria cannot control the ion channel flux anymore. Now everything becomes equilibrium. When things become equilibrium, it's now become dysfunctional. It cannot function anymore, right? So sometimes you have to have active transport to be active. And this particular antibiotic, plays a big role going and disrupting the active transport, so that so everything in the cell become equilibrated, and because it become equilibrated, you cannot, or you can no longer function. Okay? And that's pretty much it I have for lecture. All right. So if you have any questions, let me know. Otherwise, go ahead and take your break. We'll resume back at 5:30. And we'll do a Kahoot review. And after we're done with the Kahoot review, we're gonna play a kinetic game. There's no lab today, or no wet lab. We're going to do a mental lab today, using paper airplane. All right? And the winning team gets extra credit. Yeah.
You're going to explain how to do it?
Don't worry.
Yes, I know.
gonna punch up a paper ball and something like that. Huh?
Today. So today, when you guys did a paper.
We're just gonna use them and play a game.